I'm introducing myself because you all know me. I'm Bess. First I'd like to thank the Nathan Schock Center and the School of Medicine Women's Health pilot funding programs for supporting this work. So what I'm going to tell you about today was all funded by this particular pilot program. I'm not going to be talking about other things that are going on in my lab right now, just this one project. I'm going to hit the lights because I have a lot of imaging. Okay. Okay, so if you've heard me present before, you've seen these pictures. This is Alwa Alzheimer and his patient, Augusta Dieter, who he met in the early 1900s. She was the first person diagnosed with Alzheimer's disease. After she died, Alzheimer performed histopathological staining of her brain and found what he described as plaques and tangles that we hear about all the time today. So it was first described in 1906, but it wasn't until 1976, so 70 years later, that it was actually recognized as a really common cause of dementia and major cause of death. So 70 years went by without really much work on Alzheimer's disease. So 20 years later, the first FDA-approved drug came out for the treatment of Alzheimer's disease, donepezil, in 1996. It's a cholinesterase inhibitor. All of the subsequent drugs that were FDA approved are either cholinesterase inhibitors or NMDA receptor antagonists. None of these work really well in all patients. They don't work for an extended period of time. They're not disease modifying, so they're not actually designed to target the disease process, and they don't delay disease progression. So currently, Alzheimer's disease is the only disease among the top 10 causes of death in the United States that lacks a therapy to stop, slow, or prevent the disease. So my argument is that in order to rationally design drugs that are going to target the disease process, we have to understand the disease process. We have to know how you go from a normal brain to an Alzheimer's disease brain. We know that tau accumulates and amyloid beta accumulates in the brain. So these tangles and plaques that Alzheimer first described, we know are made up of tau protein or amyloid beta protein. And because of a genetic association between the amyloid precursor protein in Alzheimer's disease, there's been a historical focus pharmacologically on amyloid beta. And clearly amyloid beta is very, very important and central to the disease because of this genetic linkage and all the other science supporting this. However, it's pretty clear at this point that targeting a beta is not going to be the best way forward because people have tried and tried and tried and tried, and there have been such failure in all the clinical trials. So because of this, and because tau also accumulates in many other neurodegenerative disorders, and it's the primary pathological feature of these disorders, my lab chooses to focus on the tau protein. So our big question is, how does pathogenic tau cause neurodegeneration? What are the steps in the cell? What's the toxic cascade connecting pathological tau to neuronal death? And the systems that we use are primarily a fly model of tauopathy, which most of you have heard me talk about a lot. I'm not going to introduce the model very much. If you have questions, you can ask. Post-mortem human brain, and we also use mouse models of tauopathy that we get from collaborations. The story today that I'll tell you about is mostly a fly study plus some human. Okay, so before joining UT Health and the bar shop, I had discovered this cascade of cellular events that link tau to neuronal death. So first, others had described in multiple model systems that tau causes an overstabilization of the actin cytoskeleton and actin bundling. So you get actin filaments and they bundle up. So I found that the bundling of filamentous actin affects this complex that's in the nuclear envelope that links the actin cytoskeleton to the lamin nucleoskeleton. So inside the nucleus, you have this coating of lamin filaments on the internal surface. So overstabilization of filamentous actin decreases lamin protein levels and causes, got a movie coming, causes invagination of the nuclear envelope. So you can see in the movie here we have a control on the left and Alzheimer's disease on the right. This is the nucleus from patient material. On the left, the nucleus is nice and spherical, like you normally would see in a textbook. On the right, the Alzheimer's disease nucleus has these invaginations of the nuclear envelope. You can see here and here as you zoom through. So all of this data that I'm showing you as I'm going through is from human, but I used the fly to show that each node in the sequence of events was actually caused the next event to happen, and that it actually mattered for neuronal death. The involution of the nuclear envelope and the loss of that lamin nucleoskeleton causes a decondensation of that really, really tightly wound up heterochromatin that normally tethers to the nucleoskeleton. So in control versus Alzheimer's disease, again, here's the lamin nucleoskeleton in control. In Alzheimer's, you see this invagination of the nuclear envelope. And then I've also stained for a protein that's a marker of heterochromatin. So in the control, it's nice and bright. It rings the nucleolus. In Alzheimer's disease, there's a significant reduction of this heterochromatin staining. You can see this also by Western blot. This is a marker of heterochromatin that I'm using here, is decrease in the patient material as well, in the neurons. This causes expression of genes that are normally silenced by heterochromatin. So when you have that DNA that becomes more open, it allows transcription of genes that are normally silenced by heterochromatin. So comparing gene expression between Alzheimer's disease and human control, genes that are normally silenced by heterochromatin, about 30% of them are increased in the Alzheimer's disease brains, demonstrating that heterochromatin relaxation has a big impact on gene expression. And then this aberrant gene expression causes activation of the cell cycle. This is fly data. This is a beta-gal-laxi-based reporter of heterochromatin relaxation. So this is the fly brain. In control, you don't have any reporter activation. There's no heterochromatin decondensation. You don't have any cell cycle activation based on PCNA staining. However, in the tau-transgenic flies, you have heterochromatin relaxation, which co-localizes really nicely with activation of the cell cycle. So neurons are normally post-mitotic. They're not dividing, except neural stem cells. For the most part, neurons aren't dividing. However, in the context of tauopathy, you get this aberrant activation of the cell cycle. You can see this also, so control versus tau-transgenic fly brains with staining against phosphohistone 3, which is another cell cycle marker. This is specific to neurons because we co-stain with the antibody against LAV, which is neuron-specific, and see that it's only happening in the neurons. Okay, and this drives neuronal death. So I did this whole cascade, and when I first started here, I did a Texas Research Park seminar and got some tough questions, particularly from Yuji, who's not here now. I don't think he's here, no. Asked some tough questions about, I have this whole sequence of events, but for things down here, does targeting them pharmacologically, is it really going to matter for people? It's so late in the disease course. And I get that question. I had genetically manipulated, these types of things and saw that it suppressed tau neurotoxicity, but I totally understood his point. And so at that point, I took a step back and revisited some observations that I had made in the past. When you're reading through papers and you notice something and you say, and then you kind of forget about it, and then you keep reading and focusing on what you were originally doing. So I thought about this again and realized that these 4 nodes in the cascade of tau-induced neurotoxicity have a lot of overlap with less differentiated cell types, stem cells, and de-differentiated cancer cells. So there's a whole literature to support this. I'm just going to show a few pieces of data to tell a story. So this invagination of a nuclear envelope, this is a paper from 1997 showing in neural precursor cells in the hippocampal subventricular zone. Here's one with a nice nuclear envelope invagination here. You don't see these very often in neurons in normal neurons. Heterochromatin relaxation. This is some data from a 2008 paper. So this is just an example, but there's much more data to support this idea, is that, so in embryonic stem cells, this is where the heterochromatin is within this area of the genome, so just a little bit. If you differentiate these embryonic stem cells, you get a lot of heterochromatin formation in this area. in liver tissue or brain tissue, you also, fully differentiated, you also have significantly more heterochromatin than you do in these embryonic stem cells. So the authors conclude that embryonic stem cells versus, fully mature neurons, the DNA within the nucleus is more open in the embryonic stem cells, and it's not tethered to the nucleoskeleton, the laminonucleoskeleton. But in the brain, you get more tethering to the laminonucleoskeleton and more heterochromatin formation as neurons differentiate. You can imagine a less differentiated cell type versus a mature neuron. There's a lot of transcriptional changes that happen there. Clearly, in cancer, there are a lot of transcriptional differences between, normal cells and cancerous cells. Some data that I had gotten in 2014 was comparing the transcriptional profile between different types of brain tissues and doing principal component analysis to see which of the different types of tissues were most similar to each other. So the data sets that we were working with were laser captured neurons from human Alzheimer's disease or control, and then total brain homogenate from control, incipient AD, moderate Alzheimer's disease, and severe Alzheimer's disease, and then fetal brain homogenates. So the first principal component that couldn't be explained just because it was in the same experiment was principal component 4, which groups the Alzheimer's disease, laser-captured hippocampal neurons, the severe Alzheimer's disease, and the fetal brain in the same principal component, suggesting that the transcriptional profile among these three types of tissues is more similar to each other than to the other types of tissues. So this brought me to to think that perhaps all of these nodes in the cascade actually reflect the fact that neurons are losing their terminally differentiated state. So at this point, Adrian joined my lab. He's in the CGM program in biology of aging. He's supported by the T32 training grant, and this project is supported by the Nathan Schock Center. And he set out to test the hypothesis that tau disrupts the cellular program that actively maintains terminal neuronal differentiation. So I learned from Adrian that neurons, once you fully differentiate into a neuron, you don't just get to rest there. There's a active cellular program that maintains that state. And if you lose that state, you lose the terminal, you don't de-differentiate all the way back to a stem cell, but you can lose some of those mature neuron phenotypes. So when Adrian first joined the lab, he was making these long lists of genes of proteins that he thought could regulate this process. I learned about proteins that are called terminal selectors. So these are proteins that maintain terminal differentiation. One of the first experiments that he did was just to use a marker of terminally differentiated neurons that a lot of other people had used in the literature. This is in tau transgenic versus control flies at day 10 of adulthood. So fasciculin-2 is a cell adhesion molecule. It's expressed in differentiated neurons of the mushroom body of the fly, which is an area that's important for learning and memory. But it's not expressed in neuroblasts or young neurons that are adjacent to neuroblasts. So this is the border of the top of the fly brain here. These are the mushroom bodies. And you can see nice FAST2 staining here. However, in the tau transgenic flies, basically you can't see this FAST2 mushroom body staining anymore. So that was his first piece of exciting data. Then he started thinking about these terminal selector proteins. And he became really, really interested in one called Prospero. Prospero is a terminal selector that's a binary switch for cell fate. So when you have high levels of Prospero, the neuron is fully differentiated. It's a transcription factor. It silences stem cell-related genes and cell cycle genes, and it increases expression of terminal differentiation genes. When you have low levels of Prospero, it maintains a STEMI state, so stem cell-like fate. It increases stem cell fate genes, increases cell cycle genes, and decreases terminal differentiation genes. So he was giving lab meeting one day. He was saying, I think Prospero is really the central regulator. I think it's really, really important in tauopathy. He hadn't done any experiments at this point, but he was really enthusiastic about it. The next day, we get back the RNA sequencing data from control versus tau transgenic flies. And what do you know? The most significantly differentially expressed gene in the control versus the tau transgenic flies was Prospero. And I said, this will never happen to you again in your entire life. Like, this is the most amazing piece of data that I've ever seen. And if he had analyzed it himself, I wouldn't have believed it for a second. But he didn't analyze it himself. Hanye and Habil at Texas State analyzed this data. So we were really excited and thought that we might really be onto something that could be true. I also want to point out so these are only the top 10 decreased versus increased transcripts in the tau transgenic flies, but I want to point out Staufen as well. I'm going to come back to that one later. It is a double-stranded RNA binding protein that binds to Prospero and regulates Prospero mRNA localization within the cell. All right, so to see if Prospero depletion can also be detected at the protein level, he did a Western blot. At day 1 of adulthood, this is a time when we don't see any neuronal death in the tau transgenic flies. He doesn't see any significant decrease in Prospero levels in control versus tau transgenic flies. However, when he ages them to 10 days, there's a significant decrease in Prospero here in the tau transgenics. The RNA-seq data was done at 10 days as well. And there's a fair amount of neuronal death at that time point. An appreciable amount of neuronal death at 10 days, but not overwhelming, like it's really late-phase disease. When he looked by immunofluorescence, he also could detect the decrease, and it was more striking, really, by immunofluorescence. So this is a whole Drosophila brain here that you can see with a DAPI. A little bit of it's cut off the edge. And this is the Prospero staining. So it's in what we call the central body of the brain. However, in the tau transgenic central body, you can see a reduction in Prospero staining. When he looks at higher magnification, he also sees a difference in localization of Prospero staining within the nucleus. So while it's pretty diffuse in nuclei of control fly brains, it's more punctate in the tau transgenic fly brains. And we don't know what this means at this point, but it's an interesting thing to note. Okay, so at this point he asked, we know that this protein that's a central regulator, a terminal selector that maintains terminal differentiation, is decreased in tau transgenic flies. When is this happening? Where does it lay in this cascade of toxic events? So he first asked, he used some genetic tools that I had already previously used in my previous publications to relax heterochromatin, to induce nuclear envelope invagination, and to affect the actin cytoskeleton, and asked if any of these affect Prospero levels. So what is the cause of Prospero depletion? So first here, he is inducing heterochromatin relaxation. This is not in tau transgenic flies, it's just in normal flies. So decondensing heterochromatin, and he does not see a significant decrease in Prospero levels in the context of heterochromatin relaxation. Then he manipulated the lamin nucleoskeleton, so this causes invagination of the nuclear envelope, and again, he does not see any change in Prospero protein levels. However, when he overstabilizes filamentous actin, which I told you was the first event in this toxic cascade of tau-induced neurotoxicity, he sees at 1 day no difference in Prospero levels, but if you age the flies to 10 days, he sees a significant reduction in Prospero staining, in Prospero protein levels, just by genetic overstabilization of filamentous actin. And he also sees that same punctate staining pattern of Prospero in brains of the tau transgenic flies. So control, fairly diffuse, and then Prospero, fairly punctate. It looks more punctate on my screen than it does up here. Okay, so we thought if Prospero is this master regulator of cell fate, and if it maintains a terminally differentiated state in neurons, you would expect to see other genes that are regulated by Prospero should also be affected in the tau transgenic flies. So we took our RNA sequencing list and analyzed it to identify all of the genes that are regulated, that are known to be regulated by Prospero. And so he saw many genes here that are known Prospero targets that were differentially expressed in our tau transgenic flies, suggesting that it's not something that's just Prospero, rather it's a whole cellular program, maybe regulated by Prospero, that's disrupted in the tau transgenic flies. Staufen, I mentioned earlier, it was in the top 10 most depleted genes. So it is a double-stranded RNA binding protein that regulates, that's a known regulator of Prospero. So he also took our RNA sequencing list to see if there were any other known targets of Staufen that were differentially expressed, and he saw that there were. So you already know Prospero here, and these genes here. Okay, so we wanted to know if this was relevant to human Alzheimer's disease before he took it, much farther. So he took these targets of Prospero and targets of Staufen that are differentially expressed in tau transgenic flies. So there were 52 total. He determined which of them had human homologs. There were 43 of the 52 are conserved in humans. And then he went to a data set that was microarray data from laser-captured hippocampal neurons from control versus Alzheimer's disease to see if these conserved human homologs are differentially expressed in human Alzheimer's disease as well. And he saw that 12 of those 43 were also differentially expressed in the context of human Alzheimer's disease, including Prospero. This is the human homolog of Prospero. So suggesting that we're on the right track in the fly and that it is probably relevant to the human disease. Okay, so what we typically do to figure out if something is causal for neurodegeneration is if there's a protein that's depleted like Prospero, we would increase it and see if it suppresses tau neurotoxicity, and then we would decrease it and see if it enhances tau neurotoxicity. And if it does, we can say that depletion of Prospero is important as like a causal mediator of neuronal death. As you may expect from something that's a terminal selector, that's a central, that's so central to maintaining neuronal identity, whenever he decreases Prospero or increases Prospero genetically, it's toxic, so there aren't any progeny. He has some creative ways to get around this problem that he's working on now. But in the meantime, he started looking at, he got some flies where he could genetically manipulate these targets of Prospero that were differentially expressed in the tau transgenic flies. So I'll show you some of that data. So for instance, atypical protein kinase C is regulated by Prospero. It's increased in our tau transgenic flies. It's a serine-threonine protein kinase involved in cell polarity in neural precursor cells, and it regulates cell proliferation. So when he reduces alpha PKC levels in the tau transgenic flies by RNAi, he sees significantly less of that aberrant cell cycle activation that tau normally causes. He's also looked at some genes that are known to be regulated by Prospero that are not differentially expressed in the tau transgenic flies, such as nerfin-1. Nerfin-1 is regulated by Prospero. It's a transcription factor that prevents reversion of neurons into neural stem cells. So when he decreases this protein that's important for maintaining neuronal identity, he sees more cell cycle activation, which we know is toxic, than what you see with tau expressed alone. He's also taken a pharmacological approach to manipulate Prospero protein levels. There was a paper showing that leptomycin B, which is an inhibitor of exportin-1, causes nuclear retention of Prospero. So Prospero acts in the nucleus because it's a transcription factor, and treatment with leptomycin B keeps it in the nucleus where it can do its job. So we treated flies with leptomycin B. He sees, in terms of Prospero levels, that he can restore Prospero levels just by treating with leptomycin B. And then when you look at toxicity, we're looking at neuronal death here, tau plus vehicle, there's significantly less neuronal death when you treat with leptomycin B. So anyone who's heard me talk about our RNA export story, this may sound familiar because we're also using leptomycin B as an inhibitor of RNA export. So leptomycin B does a lot of stuff. I don't know what's more important, keeping Prospero in the nucleus or decreasing RNA export. Okay, so taken together, we've revised the model. So we think that pathological tau causes actin stabilization and disrupts the lamin nucleoskeleton. This causes a reduction of Prospero And all of the, not all of the, but many of the genes that it regulates, a whole cellular system that maintains terminal differentiation. And this causes loss of terminal differentiation, which causes all of these events that are associated with less developed cell types. So right now, Adrian is working on determining if reducing Prospero is, in the absence of tau, is sufficient to cause all of these downstream events. And then he's also using different drugs that he's identified as potential maintainers of terminal neuronal differentiation to see if he can suppress tau neurotoxicity. Okay, so pretty much everything was done here by Adrian. Garrett Cornelissen also, he did the leptomycin B experiments. Hanye and Habil analyzed the RNA-seq data. Thank you again to the Nathan Schock Center pilot and the School of Medicine Women's Health pilot and the training grant, NIA training grant for Adrian and NINDS for my funding.